Today on In Your Face Anatomy, we are talking about the macromolecules. We're talking carbs, lipids, proteins, and nucleic acids. Let's get into it. Okay, our macromolecules. This is chemistry part 4. Last time we talked about the pH scale. Now we're picking up with our macromolecules. All right, so with our macromolecules, that is going to bring us into the difference between organic and inorganic. So with organic chemistry, we are really talking about carbon-based compounds. So carbon compounds are almost all organic. Inorganic is kind of everything else. We'll say anything else. Anything else that's not carbon-based is typically inorganic. We do have to make the exception. There are some carbon compounds that are inorganic, like carbon dioxide, for one, is inorganic. But that's more chemistry than we care about for anatomy class. So what we want to know, organic chemistry, we're talking carbon compounds. Inorganic substances are going to be everything else, anything that's not carbon-based. So we've got 4 major ones: carbs, proteins, lipids, and nucleic acids. So that's one of the first things we want to go, what are the macromolecules? We got carbs, lipids, proteins, and nucleic acids. Nucleic acids. All right, so that would be one easy test question. What are the 4 macromolecules? So we're going to talk about each of these individually. We're going to start with carbohydrates. So with carbohydrates, what we want to know, carbs are your sugars. That's what we're talking about with carbohydrates. We're talking about all of the sugars and how we break these sugars down to make energy. That's super important. So that's their primary function. They provide energy to cells. That's what we're using. Glucose primarily we use in our citric acid cycle to turn it into ATP. Glucose is a carb. It is a sugar. So it provides energy to cells, and it is water soluble, meaning it dissolves in water. You put sugar in water, stir it up, the sugar disappears because it has dissolved in the water. So a couple little tricks for carbohydrates to be able to identify them. One is if you're given the molecular formula, we've got a ratio of 2 to 1 for hydrogen to oxygen, meaning I have exactly twice as many hydrogen as I do oxygen. So we can look at glucose for that. For glucose is C6H12O6. So I've got 12 hydrogen and I have 6 oxygen. I have exactly twice as many hydrogen as I do oxygen. That is a 2 to 1 ratio. So I know that is a carbohydrate. I could do like C11, H22O11. And again, I have that 2 to 1 ratio, twice as many hydrogen as I do oxygen. So I know that's a carb. So that's the easy way to find it if you're given the molecular formula. You're not always given that, though. Sometimes you're just given the name. And so the other trick with with our carbs, is that they end in ose. End in ose, O-S-E. End in ose. That's our other key for carbs. And so we'll get things like glucose. Glucose. So we see that ose there. That ose is a giveaway that this is a carb. We could do maltose. We see that ose. That's a giveaway. This is a carbohydrate. So that 2 to 1 ratio tells us it's a carb. And if it ends in ose, that's the other way we know that we're talking about carbohydrates. The building block for carbohydrates are monosaccharides. So we start, mono just means one, saccharide means sugar. So this is single sugars. That is the building block. We're going to take these single sugars and we're going to build more complicated things as we go. So we start off with our monosaccharides as our building blocks. If we pair a couple monosaccharides together, let's say we do 2, now we call it a disaccharide. That just means 2. So now we got 2 sugars. We put a bunch of these monosaccharides together, and that makes a polysaccharide. Poly means many, so many sugars. That is a big, complicated molecule. Under the carbohydrates are going to be the polysaccharides. Now, there's just a couple of polysaccharides that I want us to be familiar with. So let's get into those. So our polysaccharides to know. First is cellulose. Cellulose is a plant polysaccharide that we as human beings cannot digest. It passes through our digestive system. We refer to this as dietary fiber. So cellulose is dietary fiber. It just passes right through us because we don't have the mechanisms to break it down. Starch is a plant polysaccharide that we can digest. So we're eating those potatoes, plant polysaccharide we can break down. And then the last one is glycogen. So glycogen is a polysaccharide that we as human beings make in our body. We store it in our skeletal muscle as glycogen, which is easily transferable to glucose, and back and forth. We can go both ways with it. So if we need sugar for our muscle contractions, we've got glycogen kind of already there, ready to go. We also store some of it in our liver. So that's carbs. We want to absolutely know the 3 polysaccharides. There's only 3 that we're worried about with carbs. We want to know how to identify them with that 2-to-1 ratio or with OS. And we want to know they're water soluble and provide energy to cells because that's something that makes them distinct from our lipids. And we want to know that the building blocks of our carbohydrates are monosaccharides. So that's carbs. That's our first macromolecule. Now let's roll over into lipids. So with our lipids, we can also use these guys to provide energy to cells like we did with our carbohydrates. But lipids are not water soluble. They do not dissolve in water. We've all heard that phrase, water and oil don't mix. Well, that holds true with lipids because lipids are our fats and our oils and steroids. Fats, oils, and steroids. This is what we're talking about when I say lipids, primarily fats. So one way to distinguish them, if we're given the molecular formula, is to also look at that hydrogen to oxygen ratio like we did with our carbs. But our carbs was exactly 2-to-1. With our lipids, it's a much smaller ratio. So it's going to be like 15-to-1 or 12-to-1 or 8-to-1, something where we've got a lot more hydrogen than we do oxygen. So we can do, I have the example here of, we'll do C, 12H, 64O3. So we got a lot more hydrogen than we do oxygen. That is a giveaway that this is a lipid that we're dealing with. Now we have 2 building blocks. We've got glycerol, and fatty acids. Glycerol and fatty acids. So we're going to use these components to build all of these more complicated lipids that we're about to talk about. All right, so we just said that glycerol and fatty acids were the building blocks for all of our lipids. So let's take a little closer look at the fatty acids here real quick. So one thing we have to know about fatty acids is that they all start with a carboxyl group is what it's called. A carboxyl group, and that's nothing but a carbon that's double bonded to an oxygen and then bonded to another oxygen and a hydrogen at the other end. So it ends up looking like COOH. So you may see it like this, where the carboxyl group is drawn out for us, or it just may be more in a molecular formula style where they just list out the COOH, and we have to know that's a carboxyl group. But if we see that, we should think, oh, that's probably a fatty acid. What the giveaway is, though, that we're dealing with a fatty acid is this long chain of carbon atoms. This long chain of carbon atoms is key for fatty acids and what type of fatty acids that we're dealing with. So we have 2 types. We've got saturated fatty acids and we have unsaturated fatty acids. So what's the difference? Well, saturated fatty acids get their name because every possible place we can put a hydrogen, we do. So when we say saturated fatty acid, what we're really saying is this atom is, or this molecule is saturated with hydrogen. And that's why it's saturated, because we put a hydrogen every possible place. Now we know each one of these carbons has to form 4 bonds. Carbon has to form 4. So I've got 2 on this first one. I'm going to add 2 there. Now I'm up to 4. Up to 4, and add the third one there to get that one up to 4 bonds as well. And now I come back, and every place I can, I slap in a hydrogen. Now this atom is completely saturated with hydrogen. This is a saturated fatty acid. What else we want to know about saturated fatty acids? One, we are talking about animal fats. primarily with saturated fatty acid. We're dealing with animal fats. And these animal fats are typically solid at room temp. Like butter. You can take butter, put it out on the counter, and it keeps its shape because it's a saturated fatty acid. It's made from cow milk, so that is an animal fat. So saturated fatty acid is solid at room temperature. My unsaturated fatty acids just mean that somewhere along the line here in my chain of carbons, I've got at least one double bond in between the carbons. Now, it doesn't count for this carboxyl group when we draw that in because the carbon is double bonded to an oxygen. I'm looking for a double bond somewhere in my line of carbons. So right now I've kind of got it set up for a saturated fatty acid. I can draw in all of my hydrogen again, hydrogen, 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 hydrogen. So now it's a saturated fatty acid, but we want it to be unsaturated. So I can pick any spot here. I'm going to go right in the middle and add in a double bond. Now I've got a double bond in between my carbons. So these two carbons now have 5 bonds. They're not allowed to have 5. They have to have 4, which means we have to get rid of a couple of these hydrogen. I'm going to get rid of those two just kind of at random. Let me get rid of this saturated 2 so we're not confused by that. So as soon as I have that double bond in there, I know this is a unsaturated fatty acid. Unsaturated fatty acid. And because there's only one, then that makes this a mono unsaturated fatty acid because there's only one double bond. But if I add in another double bond, boom. And we have to get rid of some more hydrogen to keep our bonding because carbon's only allowed to have 4 bonds. Now they all have 4. But I've got 2 double bonds, so that makes this now a poly unsaturated fatty acid. So a mono is just 1 bond, anything more than 1, and it becomes a poly unsaturated fatty acid. So with my unsaturated fatty acid, these are typically plant fats. Plant fats, but we know these better as oils. Oils. So this is like peanut oil or olive oil or canola oil or corn oil. Any of those oils, they are always liquid at room temperature. This is an unsaturated fatty acid. They're derived from plants, they're liquid, and it's the double bonds that give it away. Remember, just checking that line of carbons to see if it's saturated or unsaturated fatty acids. Next up, we've got our triglyceride. So a triglyceride is just a single fat molecule. Is what it is. So we take a glycerol, which is one of our building blocks for lipids, and we combine it with 3 fatty acids. So tri is the key there. Tri for 3. I've got 3 fatty acids on a glycerol. There's both my building blocks to make a single fat molecule we call a triglyceride. Probably the most important one, I'm going to put a little star in front of it because you should as well, is the phospholipids. The phospholipids we talk about in this chapter, we come back to again in the next chapter when we talk about the cell, because our phospholipids we want to associate with the cell membrane. The cell membrane, super important part for the cell membrane, the phospholipids. So with a phospholipid, we have a glycerol with 2 fatty acid chains instead of 3. We just have 2, but we also have a phosphate group, and that's important. So when we start putting our phospholipids together, this is typically how we're going to draw it out. This is my phospholipid molecule. So the head part of it here, this is what's made out of the phosphate group. So there's my phosphate in the head part of my phospholipid, and I've got these 2 chains, 2 tails here hanging down. These are my fatty acids. So the thing that makes phospholipids so interesting is I've got this one molecule here. Got the phosphate part, got the fatty acid parts. The phosphate part we call hydrophilic. Hydrophilic because it loves water. It loves water, wants to always be around the water, be immersed in the water, but just the phosphate part. The fatty acid tails, on the other hand, we call hydrophobic. And it hates the water. It hates the water. Doesn't want anything to do with the water. So that makes it interesting in how our cell membranes come together, because we have half the molecule that loves water, half the molecule that hates water. And that's a phospholipid. It both loves and hates the water at the same time. So that's what makes phospholipids interesting. Be up on that. Make sure you understand what makes the head of a phospholipid, what makes the tail. Is it hydrophilic? Is it hydrophobic? Does it like water? Does it hate water? And all of that comes back to that picture of my phospholipid. All right. The last lipid we care about is steroids. So steroids, complex connected rings of carbon that are all bonded together. We can see kind of a ring structure that I've drawn in. We just have to know that at every bend here, there's a carbon and all of its little associated structures. That's something you just got to know. But this is the only thing I'm going to come at you with a ring structure. So if we see anything that looks anything like this, we should know that's a steroid, and steroids are lipids. All right, our third and most complicated macromolecule are the proteins. So proteins, we will talk about proteins from now until the end of Anatomy 2. They have a variety of functions. Proteins do a little bit of everything. We use them as enzyme proteins to help speed up reactions. We use them as antibody proteins to help fight off invaders into the body, whatnot. Tons of functions for proteins. Our cells use them constantly. So with proteins, most of the proteins are going to end in the suffix -in. That is a loose rule there, where like the os for our carbohydrates is pretty solid. Anything that ends in os is going to be a carbohydrate. With -in as our suffix, that's most of the time it's proteins, not all the time. We do have other classes of proteins like enzyme proteins. They end in -ase. Don't confuse that with os. Os was the carbs. -ase is an enzyme, and enzymes are proteins. So the building blocks of proteins are amino acids, super important. Building blocks of proteins, amino acids. And we have 4 parts to a typical amino acid. We have the carboxyl group. So that's what we looked at earlier with our fatty acids. Carboxyl group, that's COOH. We also have an amine group, or an amino group, which is NH2. We have a single central carbon that's bonded to a hydrogen, and then we have the R group. So what that ends up looking like is there, I got a central carbon, boom, with my COOH. I've got an amino group over here. I've got my carboxyl group over here, and then I have the R group. So the R group just means the rest of the atom. It's everything else. It's what makes this amino acid different from the next amino acid over to it. It all comes down to the R group. So all of my amino acids are blessed with this configuration. I've met all of my bonding requirements, so they all come with this. But it's the R group that makes each one different from the next. All right, a protein's shape is everything to a protein. We call it shape, it's conformation. Protein shape is its conformation. And why it's so important, because proteins have to fit into receptors of a specific shape. So it's got to fit into the receptor. If that protein doesn't fit into that receptor, then that protein's useless for us. It's not going to do anybody any good. So protein shape is its conformation. There are things that can cause a protein to lose its shape. And when that happens, we say the protein has denatured, or we're going through the process of denaturation. You may hear something along those lines. And that just means the protein has lost its shape. Things like high temperatures can cause a protein to lose its shape. That's why you feel like butt every time you're sick, because if you're running a fever, your antibody proteins have unraveled and they're not able to fight back. The temp has got too high. pH changes can cause the protein to lose its shape. Radiation, certain chemicals, all of that can cause the protein to lose its shape. Peptide bonds. These are the bonds that hold a protein together, essentially. Let me not say that. Let me say these are the bonds that we find in between amino acids. Now, we just said amino acids are the building blocks of protein. So as we start to put together a protein, we're doing this amino acid with this amino acid, and they're bonded together with a peptide bond. So that's the 5th kind of bond we need to be familiar with. We did hydrogen, ionic, polar, non-polar, and now peptide bonds we want to know. So if we wanted to build a protein, we need to understand the protein structure, or how it's done. So it starts out with the primary structure of a protein. And this is simply the sequence of amino acids that we string together. I could take this amino acid and then this amino acid and then another one of these and another one of these and then one of these over here, or maybe an amino acid from way back here. And they're all get bonded together in a specific sequence. That is my primary structure. It's held together by our peptide bonds. And one of the key ways is to remember is it's like beads on a necklace. If you're a little kid, you ever put beads on a necklace, one bead and then the next bead and then the next bead. That's kind of how the primary structure of my protein gets laid out. Once we get that all figured out, we move into the secondary structure. And the big thing with secondary structure that I want you to be familiar with, one, hydrogen bonds only. We're only using hydrogen bonds with the secondary structure of my protein. What this does is it allows some minor bending and folding to occur. So we really get two shapes that arise from the secondary structure. We get the alpha helix, so that's the little alpha sign. So we get an alpha helix, kind of looks something along this. There's an alpha helix. And then we get the beta, we got the elongated B, the beta pleated sheet. So beta pleated sheet is kind of folding back and forth, back and forth, back and forth. So we get these two shapes from my secondary structure with some minor folding going on. Then we roll over into my tertiary structure. So my tertiary structure is just a larger level of folding that occurs. Now we're folding entire alpha helixes and entire beta pleated sheets around each other to give us this really complicated shape. It's held together through hydrogen bonds again, but now we're also doing covalent bonding. Once we get that shape figured out, we roll into the quaternary structure, and this is our finished protein. So this could be like hemoglobin or actin or tubulin, something along those lines. It's our finished protein that we're done is our quaternary structure. So know what happens at each level of my protein structure, and one of the key things to distinguish is the bonds. I've got peptide bonds for primary, hydrogen bonding only for secondary, hydrogen bonding and covalent bonding for my tertiary structure. So that's an easy way to make that distinction between them. All right, our last macromolecule is the nucleic acids. So nucleic acids, they have a building block called nucleotides. Nucleotides are the building block for nucleic acids. They are made up of carbon, hydrogen, nitrogen, oxygen, and phosphorus. That's going to be key here in a little bit. Phosphorus. It's our only macromolecule that really comes with a P in its molecular formula. So that's how we're going to make that distinction. So we have just two nucleic acids to be familiar with. We got DNA. So we want to know, what does DNA stand for? It stands for deoxyribonucleic acid. Deoxyribonucleic acid. And typically we get that double strand of DNA that gets twisted into a double helix kind of pattern like that. What does DNA do? DNA stores the information. It keeps all of the information on how to make proteins specifically safe inside of the nucleus. We don't want anything else getting to that information because proteins are so important. We're going to protect it. So we hide the DNA away in the nucleus. There's only a one way in and out, and that's where we're going to keep the DNA to keep it safe. RNA, ribonucleic acid, uses the information. It gets in, makes a copy of the DNA, and then it runs back out of the cell and actually uses that information to start making protein. So DNA stores the information, RNA uses the information. All right, so that's a quick rundown of the macromolecules. Good luck.